Welcome to our webinar series for professionals.
So it's time to do a webinar. It's one of those Thursdays again. So welcome to you all and thank you for signing up. And we're really excited because we're launching this new DNA gut test. We already have a lot of tests and a lot of experience with gut tests, of course, but this was really like a missing piece in the puzzle, you can say, to get this genetic insights. And we've been running lots of tests here in the background to get lots of experience and questions so that we're prepared to questions from you and especially Helen, of course, who's gonna educate now, who's like the master behind the test and who's been looking at all the research work and looking at all the clinical insights that there is in relation to the genes that we're looking at. So she's been especially very, very busy. And she's in Copenhagen now, actually sitting in the head office, so not in South Africa. So, she will also be in London at the event that we have in the middle of November, 14th to 16th of November in London. Lots of people are signed up already, but there's still room for more people. So you will meet Helen there if you show up together with other presenters. So it's going to be a real exciting crowd to learn from for sure. And then we have here another webinar on the 23rd of October in regards to hormones, but of course we will send out invites about that. So enough from me here on this side. And let's, because you're all excited to hear about Helen and DNA gut, so I'll let Helen take over the stage now. And remember, ask questions in the chat. I'll take notes of them if I can't answer them right away. And then I'll take some of the questions up at the Q&A session at the end. And also we are recording so that we will share the recording with you, and Helen will also share the slides. So we will send out the slides to you and the recording when we have put it together in an email for you. So I'll just be quiet now.
Thanks, Anne-Catherine, and hello everyone. I'm seeing some familiar faces and many new, so yeah, really excited and feel very privileged to be able to present this test today. It has been over a year in the making, and just in terms of the development, there was rigorous selection criteria, a lot of insights from many functional healthcare practitioners, so really value their opinions here in terms of providing insights to put the report together. And yeah, just a big thank you obviously to the R&D team for putting this together and all the hard work over the last year. So this is an overview, an introduction to DNA gut. And what I want to start with is a little bit of background in terms of what we're looking at, and then really emphasizing and going forward how important this test is in your functional healthcare practice, using it together with other functional tests to personalize interventions going forward and to really get such useful, valuable insights that I think would be remiss if you don't have this test in your toolkit when looking at your patient's symptoms or understanding your patient's symptoms to personalize a program for them. So starting off, we're getting right back to basics, and I'm sure you haven't seen me present on these basics in a long time, but what we are really looking at is our blueprint, the human genome. And this is the complete set of genetic instructions in our cells. It's composed of our DNA organized into our genes. This is our unique code that determines physical traits, predisposition to certain conditions, and how our bodies function. And So while our genes are not fixed, the expression is influenced by lifestyle and environment, and that is where nutrigenomics comes into play and epigenetics comes into play. So our genes are static in terms of our genotype is static, but the expression of those genes is definitely not. And it is governed by obviously our genotype and our environmental exposures and our gut microbiome. So what we are looking at with this test, as with all of our other tests, is the genotype and considering a normal or baseline genotype resulting in typical response to outcomes. And then you've got your variant genotype looking at how there might be an altered response because of environmental exposures. And understanding that, you can really put into play very personalized, targeted interventions. And then we get to the other side where we're looking at our inner ecosystem, the guests in our home. So our home is created by our blueprint, our genotype, and we want to invite our guests to stay and to be very friendly and happy and merry within our home. And this is our gut microbiome, which consists of trillions of microorganisms, bacteria, viruses, fungi, all living in our digestive tract. There are more cells than in our body, and they are not just passengers. Some are, some come and go, but it is a dynamic, active organ with vital functions. We want many different guests in our home, and so diversity and friendliness is key. And so when we're looking at the function of our gut microbiome and its impact on health, we know that the gut microbiome plays a very important role in digestion and nutrient absorption, breaking down complex carbohydrates, synthesizing essential vitamins, including vitamin K, certain B vitamins, also looking at how certain nutrients can be absorbed, metabolized, iron included in that system as well. Then, of course, and very importantly, is immune system modulation, where we know around 70 to 80% of our immune cells reside in the gut. And the microbiome trains and interacts with our immune system to support good immune regulation. And that is what we're striving to do. We then have protection against pathogens with our microbiome. So our guests in our home protect from, because they're occupying space, they protect from unfriendly people coming in, or unfriendly microbes coming in, and they also produce weapons or antimicrobial compounds that can fight the bad guys. And then we have the gut-brain axis. We have actually many axes that we speak about within DNA Gut, but one of them being in terms of function and impact is our gut-brain axis that is governed by this a very important nerve and produces, and the microbiota produce neurotransmitters influencing mood, cognition, and behavior. So that is how our gut microbiome can influence our health. And so what we also want to consider is how genes influence the gut. And I was actually reminded of a quote that came from Dr. Craig Venter. He was responsible, or one of leading now over 20 years ago, the sequencing of the human genome. And he made a statement around, obviously, and we know microbiome is so, so important, but our genotype, and our genotype is also so important, because our genes influence the structure, our home in which our microbes live. So even when we're looking at our microbiome, we need to consider how our genotype is creating that structure that then interacts with our microbiome. And so it does influence what our microbiome is going to be, and it shapes that microbiome. So our host genetics shape the environment, shapes the home. Our genes determine the structure of our gut, from gut motility, mucus production, to gut barrier integrity. we also have immune system genes. So while we have our microbiome that really plays a very important role in immune regulation, we have our own immune system genes, and genetic variations in immune response genes can influence how our body interacts with our gut microbes. And then obviously we have genes encoding many enzymes that are really important for metabolizing and breaking down nutrients. So these genes dictate the enzymes we produce, how they are functioning, and these can affect what nutrients are then available to our microbes to consume. Moving and very closely related to our immune system genes are our HLA, or human leukocyte antigen genes, which are very strongly linked to autoimmune conditions and can influence the microbial recognition. And then we have our gut epithelial barriers. So if you think of this blueprint, and part of the blueprint building the walls of our home, single cell layer, these genes play a crucial role in maintaining the integrity of the gut lining, which is vital for preventing intestinal barrier permeability, or increased intestinal barrier permeability. So then we want to know how we now understand how our genes can influence our gut, how microbiota can influence our health, but how can gut microbiota actually impact our genes as well? So it's a very complex situation that we are speaking about and idea here. But when we're looking at how microbiota impact our genes, and this is where we're looking at gene expression and nutrigenomic interactions that also come into play here, we've got microbial metabolites that act as messengers. Gut bacteria can produce a vast array of compounds, including our short-chain fatty acids, such as butyrate, our neurotransmitters, as well as bile acid, and create bile acid modifications. This and some of these metabolites can create epigenetic modifications. So these metabolites can directly or indirectly influence our gene expression by altering our DNA methylation or histone modifications. And so depending on what you're producing and what metabolites you're producing, it can either lead to increased expression of pro-inflammatory genes or decreased expression of these pro-inflammatory genes, as an example. And so when we're looking at inflammation, of course, immune system regulation comes into play here, where microbes influence the expression of our genes involved in immune responses, potentially leading to pro or anti-inflammatory states. And then we've got our metabolic pathways, where gut microbes influence the expression of genes related to lipid metabolism, glucose regulation, and nutrient absorption, and can also influence our overall weight status or obesity status. So with this in mind, over the last year of intense work with the R&D team and getting really great insights from key opinion leaders in the field, we were able to develop the DNA gut test, which is a host genetic test. What I mean by host, we're not talking about an alien, we are talking about our human. Human is the host to our gut microbiome. So the human genetic test that analyzes and reports on 26 variations in 18 genes that play a role in 12 biological pathways. The included genes are based on convincing evidence and are clinically relevant to gut health, and they provide valuable insights into how our host or our human DNA forms the framework for our gut health and our microbiome, and that interaction that then comes into play to then be able to provide in the report personalized diet, lifestyle, and nutraceutical interventions, even looking at specific pre- and probiotic support to then support these relevant pathways and help one achieve and maintain a healthy and resilient gastrointestinal tract, and then thereby health span. So just to note as well that across our DNA test offering, there are 13 of these genetic variants within DNA Gut that are unique to DNA Gut. The variations that might be seen across the other tests are very specifically were included in this test very specifically because of their association with gut health, and the insights and the recommendations provided in DNA Gut are very specific to gut health. So that's also really important to remember with the genetic variations that we are including and reporting on. So I'd like to discuss in brief the different biological pathways that we are reporting on and the associated genes within those biological pathways, starting off with the gut-brain axis, which is this bidirectional communication network influencing mood, behavior, and gut motility. And here we have our apolipoprotein E gene, which differentially modulates microbiome diversity. So you will be very familiar with this gene in many of our other tests. This is probably still one of my most exciting genes to talk about, and it has become even more so when looking at the research around the gut-brain axis and how ApoE is so important in modulating inflammation, lipid metabolism, gut-liver axis, gut-brain axis, and it actually influences microbiome diversity, the metabolites produced, and thereby influencing neuroinflammation by the gut. So a really interesting one that you will learn more about in the online course, and in London we will speak more about it as well. Then moving on to the blood group antigen secretor part, and that is where we're looking at blood group antigens that influence microbe colonization. I don't want to give too much away here, but it is where we're looking at the FAT2 gene, which determines your blood group antigen secretor status. It acts like a flagpole that you can attach a flag to, and if you have that flagpole there, it's kind of made up of sugars in a way, and so there are certain microbiota that are have developed to recognize this flagpole and can attach to this flagpole and can actually use it as a food source. And depending on your secretor status, are you potentially more at risk for certain infections, or are you potentially more at risk for having some of the more friendly bacteria not able to colonize as easily because it can't recognize that flagpole? So really important area regarding susceptibility to infections as well as inflammatory conditions. Then in terms of bacterial sensing and autophagy, we're looking at genes that encode bacterial sensors and your autophagy enzymes that are responsible for detecting bacteria and that then trigger an immune response and activate your cellular self-cleaning processes. And this is a really important area where you want a balance between tolerance and defense. And if we're looking at immune regulation, these play a very, very important role here. So we're looking at your toll-like receptor 4 and your NOD2, which are receptors that detect bacteria and their components. And then we're also looking at this ATG16L1, which encodes an autophagy enzyme and maintains intestinal cell health by regulating inflammation, influencing microbiome composition, being able to kind of be the vacuum cleaner, cleaning up dead cell debris, as well as recognizing pathogenic bacteria and being able to eat those up as well. Then we have our tight junctions, which we are looking at one of the genes that form a tight junction protein. And this is a crucial barrier for your epithelium. So your MAGE2 is the gene that encodes a protein that's a protein complex, which is very important in sealing intestinal epithelial cell gaps. And disruption in this protein, partly due to a genetic variation, can lead to increased intestinal permeability, allowing bacteria, toxins, and other metabolites to enter the bloodstream, triggering inflammation and thereby an immune response. Also very strongly related to immunity is vitamin D, and we see that there are specific receptors that we're looking at on the vitamin D receptor, specific variants we're looking at on the VDR gene. And when there are these variants, it is linked to increased inflammation as well as an altered microbiome composition. So when we're looking at the receptor, it actually has this interaction with our microbiome, changing what we are expecting to see in the gut. And obviously moving from vitamin D, and we've placed these in such an order that you can link all the kind of inflammatory immune genes and pathways together, where we know that vitamin D and inflammation play very strong, they're interconnected very closely. And we see that with inflammation, we're looking at this complex biological response to harmful stimuli, and obviously trying to avoid the chronic inflammation that is partly caused by genetic variations together with environmental exposures and triggers and these genetic variations are found in genes of IL-10, which is an anti-inflammatory gene, then IL-23R, TNF-alpha, and your TNF-SF15, which are pro-inflammatory in nature. And we see that microbial imbalances and environmental factors can lead to chronic gut inflammation, and this underlies many of the digestive disorders that can go on to have systemic effects. And then from inflammation or oxidative stress to inflammation and vice versa, we see that we can have damage to the intestinal lining that can contribute to inflammation because of high oxidative stress levels. And the microbiome can both contribute and be affected by oxidative stress. So we're looking at a gene that we have not included or spoken about before, your SLC39A8. It is a transporter of some of your minerals, specifically your manganese, which is essential for your mitochondrial antioxidant enzyme, SOD2, or manganese superoxide. And that manganese superoxide requires manganese as the cofactor. If the enzyme is not working that well, we cannot get that manganese inside the mitochondria. And so the ability for our mitochondria to manage that redox status is decreased, and that can then lead to increased risk for dysbiosis. Moving and very closely related to oxidative stress is detoxification, which plays, and this is really important in being able to detoxify harmful substances that we're exposed to on our skin, what we inhale, but also very much what we ingest. Our first line of defense is with our gut microbiota is what we ingest. So everything that we take in orally actually starts to get kind of detoxified in our gut and then moves on to our liver. So this is where our microbiome is really important in helping with detoxification. But if we have detoxification genes that are very strongly expressed in our liver as well, and we have variants in those genes that lead to a reduced capacity to detoxify certain compounds, this potentially increases susceptibility to gut inflammation or other health issues influenced by the gut environment. We then have our serotonergic pathway, where we know that 90% of serotonin is produced in our enterochromaffin cells within our gut. Serotonin, we know, is this crucial neurotransmitter in the brain and in the gut. And where we see serotonin playing a very important role in the gut is in regulating gut motility, visceral sensitivity, which is the perception of internal organ sensations, and also plays a role in secretion. And so we're looking at a receptor here, and if this receptor is really sensitive, it can lead to increased symptoms around irritable bowel syndrome. Then we have starch degradation also sometimes, and if you are not able to digest your starch properly, this can lead to symptoms associated with irritable bowel syndrome, especially the diarrhea form. And so we're looking at an enzyme here, SI, which encodes your sucrase isomaltase enzyme, and this is produced by actually both the host, so both our human body, and in fact gut bacteria. And this enzyme is responsible for breaking down starches into simpler sugars. The efficiency of this process results in and resulting byproducts influence your nutrient availability for both the host and microbiome. So depending on what is happening there, you're going to have certain energy sources that you might not want around, and that can also lead to cramping and diarrhea-related symptoms and IBS-related symptoms. Then we have our gluten intolerance, which I'm sure you're all very familiar with, where we're looking at individuals who may be genetically susceptible to gluten intolerance, celiac disease, and also these specific genotypes have been related to non-celiac gluten sensitivity. There are others also related to non-celiac gluten sensitivity, whereas the HLA-DQ2, DQ8, your human leukocyte antigen, with these specific genotypes are very strongly related to gluten intolerant celiac disease. And even if you do not develop actual celiac disease, you may still develop this sort of antigen or immune response to gluten. And then looking at lactose intolerance, we are looking at the gene encoding an enzyme that really stops in most individuals being produced around the age of 2. And so we're looking at adult lactose intolerance, or lactase persistence, which is the opposite. So some individuals are able to continue to make this enzyme into adulthood. And so we see that undigested lactose can cause gas, bloating, diarrhea, and directly impact gut comfort and microbial activity. So those are our 12 pathways that we are reporting on, with those genetic variants that then impact what we are going to speak about as our 5 DNA gut pillars. And these genetic variants can influence these gut pillars. So why we want to identify these as the pillars of DNA gut is that you can look at the genetic variants that you carry, or that your client carries, within the DNA gut report, and where they fit into these 5 pillars to address both the gene, but also the pillars of which is sometimes nice from a functional approach to see, are there microbial alterations and potential presence of pathobionts, which is where you're looking at an alteration in your good and bad bacteria, or where good bacteria becomes bad because it's too prolific in certain amounts. We also have our aberrant microbial metabolite production, and that is where bacteria is not producing enough of the good stuff, or producing some more of the more pro-inflammatory products and metabolites that we do not want around. We're also going to, we want to address gut barrier dysfunction, and that is where your gut epithelium plays a really important role, in genes related to protecting that gut epithelium. We speak about altered gut transit time and motility, and I mentioned briefly the serotonin receptor playing a role there, and your immune dysfunction, where many of these genes play a role. And of course, each pillar cannot stand on their own, and they kind of influence each other as well, but they're very specific recommendations that we can put into place to address each of these pillars, based both on the pillar and on the genes that fall within that pillar. So if there are weaknesses in any of these pillars, we can see, or we may potentially see, negative consequences on human health. And this is where the genetic predisposition plus environment plus microbiome plus exposure over your lifetime increases risk for certain conditions, from inflammatory conditions, your IBD, ulcerative colitis, Crohn's disease, as well as your IBS, celiac disease, non-celiac gluten sensitivity, lactose intolerance, H. pylori infection, and other gastrointestinal or gastroenteritises, so some of your viral gastroenteritises, your non-alcoholic fatty liver disease, cognitive decline, and depression and mood disorders. And this is where we're looking at the research, and we are able to see these genetic variations have been associated with a specific outcome. Sometimes they speak about the intermediate points, so pro-inflammatory markers or gut microbiome markers, but oftentimes they will speak about that endpoint being what I have listed here. So really important if you're looking at any of these, considering who's coming into your practice, this is something where the test would be really valuable. And obviously, even beyond that, for anyone really just wanting to improve health and look at just improving gut health and health span, this is an absolute must in your practice. So I'd like to go over the DNA gut report quite briefly with you now to show you what you will receive if you order it for yourself or for your client. So welcome. This is a no name, but welcome no name to your DNA gut because it's obviously a sample. What you will then see is a layout to present what you are expecting to receive or the important pillars that we are speaking about. Briefly to describe them, we've got our gut transit time and motility, and if that is impacted negatively, it leads to dysfunction in motility and transit time, which can also disrupt the microbiome. We've got our presence of pathobionts, and if that is highlighted, it is due to an imbalanced microbiome potentially, which allows normally harmless resident bacteria to become pathogenic under certain conditions. We have our aberrant microbial metabolite production that I mentioned, and this is where we have a deficiency of beneficial metabolites or overproduction of the harmful ones. We have our gut barrier integrity that might be compromised, and this is where an unhealthy microbiome, together with genetic variants, potentially high inflammation, leads to a weakening in that intestinal lining, allowing harmful substances to enter the bloodstream, causing a systemic inflammatory response. And then we have our altered immune function, which can be caused or driven by intestinal inflammation, and that then drives microbial imbalances leading to gut dysbiosis. So we are aiming for homeostasis within the gut, and that can be achieved by understanding what our genotype is, what our microbiome is saying, looking at further functional test markers, understanding that test pairing, and getting insights to create a really holistic plan for diet, lifestyle, nutraceutical, and supplement recommendations. Then within the report, we will see we've got our report summary priority page, which will show you the biological areas that we are able to report on. And then based on the algorithm of using your genotypes, there's an algorithm that would then give you insights as to whether the individual, your client, yourself, is a low, moderate, or high priority within some of these biological pathways, and helps you identify where you want to target your interventions. Is it around inflammation, autophagy, gut-brain axis, whatever it might be? And that is, of course, based on your genetic variations. So here we give the genotype results table in the report, again, divided according to biological area, the gene name associated with that, or that gene associated with that biological area, the genetic variation on that gene. We then give the client's result, and then the associated or assigned impact factor based on the genotype result. And that is obviously, it's qualitatively assigned, but based on what we're seeing in the research and the literature saying this specific genotype increases risk for inflammation or an altered microbiome or certain in-state outcomes such as inflammatory bowel disease. And so that is how we assign that impact factor. Obviously, also, the more malleable that gene is, how we can intervene in the pathway in gene expression, we also take that into account. So that is all given in your report, the summary and then the genotype results table. And then also in the summary pages, what I'd like to highlight is we have listed the genes according to where they impact one of these 5 gut health pillars. And so you would be able to identify when they've got the purple dot next to them, it means that there is a variation that increases risk for that pillar being weakened. And if that pillar is weakened, you would want to maybe then, from a genetic standpoint, understand that that is going to be a priority for them forever, because obviously your genes don't change. So that is something you would continually want to support. So obviously seeing the here and now with your patient, and then always keeping in the back of your mind, there may be a slightly aggravated immune response, or gut barrier integrity. The genotype is encoding for walls that are just slightly shaky. And so we always want to make sure that we need to put in a bit more maintenance with those walls. As an example. So the risk genes that may adversely affect the pillars of the gut, of DNA gut, and require extra support, as I've said, are highlighted in purple. The gray, that would be the normal genotype that does not require extra support. And then we are able to show you, based on the genetic variation, potential areas of risk that are associated with those gene variants. And those would either be shown in a color or grayed out if it is not a risk for you or your client. And then from there, from what you can see from the pillars, we then take it a step further and provide this one-page summary that we hope is as easy and summarized and succinct, but comprehensive enough to be able to address these 5 gut health pillars, where we provide targeted diet, lifestyle, nutraceutical advice, and then obviously some of the things you might need to avoid on top of that. And that is, these interventions and recommendations are summarized, but based on how we can address that pillar, but also targeted to the specific genetic variants that we see coming up in the report. So that is a very nice personalized one-page summary that you can highlight where you see your patients or your clients need a little bit of extra advice and support, and then tick where they're going right already, and that can be a step-by-step process. So this is something you can take home, or your client can take home and keep with them as a reminder of what they need to do. We then get into more of the detail around the report, and this is, I think, maybe more for most of the practitioners, but some of the clients really do like it as well, if you like your detailed, if you get your detailed clients. And this is where we are showing the description of the pathway, gene and genotype specific information, as well as the impact factor associated with that specific genotype. We then show an image for each of the pathways, and dependent on whether there are genetic variants that increase risk or show baseline risk, that image is going to be different. And we'll show how mechanistically and within the gut that specific pathway is influencing gut health and thereby overall health. And so we provide that for further insights for both you and the client. Then also providing a priority level of the pathway, low, moderate, or high, including summary recommendations based on that pathway now for diet, lifestyle, nutraceutical, and then some follow-up functional testing that would be really relevant. And we would suggest you do alongside the DNA gut test, looking at the genotype that you have and what you might want to look for further on and identify in some functional testing. We also then provide potential areas of risk. So here it's the pathobions and potentially increased infection. So that is a summary of DNA gut and what you should expect to see and why it is so valuable. I'm going to take you through some useful examples in something that you might be seeing very regularly in your practice, with symptoms related to irritable bowel syndrome and some of the genes that you would want to extract or just highlight if you're seeing them come up in DNA gut. So here, I'm going to highlight some inflammation genes, gluten intolerance, serotonergic pathway, and your starch degradation. Starting off, let's look at inflammation and your interleukin 10. So your interleukin 10, as I mentioned previously, is an anti-inflammatory cytokine. It suppresses secretion of many of your pro-inflammatory cytokines and chemokines, and is actually known as one of the main regulators of inflammation in the gut. It's very strongly expressed in your epithelium in the gut. It limits host immune response to pathogens. So we obviously want an immune response, but sometimes it can be overinflated, overexcited, and so we want it to be just dampened down a little bit, and that is where IL-10 plays a very important role. It prevents damage to the host and maintains normal tissue homeostasis. The mechanism with regards to irritable bowel syndrome is that it is critical to maintaining healthy epithelial homeostasis through supporting cellular function, your gut barrier, and mucosal tolerance. And as I mentioned, it is this master regulator of the anti-inflammatory response in the intestines. So there's really good evidence to support why we included this specific SNP in inflammation within DNA gut. It has been found to be a candidate single nucleotide polymorphism of IBS. This was actually a systematic review and meta-analysis that found from over 955 cases that the CC genotype is associated with, in fact, a decreased risk. So it depends on how you look at it. You can see CC is decreased. If you see the CT or TT genotype, you may consider, there might be something going on there that might predispose to this more inflammatory condition. And so here, it is because the CC genotype leads to increased expression of IL-10. Increased expression of IL-10, a better anti-inflammatory response. Again, another study now in Chinese patients, specifically looking at the diarrhea-predominant IBS, again, showing the same thing. So, and some of these really, these articles really recent, which is quite nice to see. And then there's another SNP that we're testing on IL-10 as well. So looking at 2 variations on this gene. And here, the other variation is now also, it's the C, but it's now on a different position, showing significantly higher, and also higher IL-10 concentration. And so this is where we see the TT and CT, so T allele, you can consider the risk, showing quite a significantly increased risk for having IBS-related symptoms, and in this case, diarrhea-related symptoms. So what was also interesting to see is those who had IBS and had the CC genotype presented higher pain thresholds, and it's potentially due to that anti-inflammatory effect. So really useful in terms of understanding insights to predisposition to IBS, but also with IBS, what would you want to consider doing if you come up with IL-10 risk variants? We want to see how we can support IL-10 expression, because remember, better IL-10 expression, a better anti-inflammatory response. So vitamin D has been shown to directly upregulate IL-10, as with an anti-inflammatory diet, of course, and avoiding nutritional deficiencies that might add to inflammation. Cinnamon extract in certain studies have been shown to also upregulate IL-10 expression, and then moderate intensity exercise. We would want to put in these interventions and then do pre and post testing with our functional and real-time biomarkers, including things like HSCRP, looking at if we can do an actual IL-10 level, and then some of our stool tests that give us markers of inflammation that would also help us to identify how well our interventions are working. Next, we're going to speak about another one that you will not be familiar with. It's a long name, your TNF-like ligand 1 alpha vascular endothelial growth inhibitor. So please don't, just ignore the name. But what we need to know about it is it's a cytokine, pro-inflammatory, that belongs to your TNF ligand family, so your tumor necrosis factor. And if you think tumor necrosis factor, think pro-inflammatory. It's abundantly expressed in your endothelial cells, but is not expressed in either beta or T cells. And the expression of this protein is inducible actually by TNF-alpha, other TNF ligands, as well as your IL-1 alpha. So if you've got high IL-1 expression, and that you might be able to see from your DNA core or DNA health tests, If you've just got overall high pro-inflammatory triggers, you are going to get higher TNF and IL-1 being produced. If you are overweight, if you have a high fat percentage, these pro-inflammatory cytokines are going to be produced, and they are going to induce this TNF-like ligand expression as well. So what happens then is, in terms of the mechanisms behind this, it can activate your NF-kappa beta and MAP kinases. It activates an autocrine factor to induce apoptosis in endothelial cells, which of course, when regulated, tightly regulated, it's important. It's really important for immune defense. It inhibits endothelial cell proliferation and may function as an angiogenesis inhibitor, but it also then may trigger an immune response to your Th17 cells and play an important role in the development of many autoimmune and inflammatory diseases if it is overexpressed, and if we've got high TNF-alpha, high IL-1, and then a genetic variant leading to overexcited expression of this gene, leading to a chronic inflammatory condition. So we see here that G variant carriers, AG and GG genotype, increases the risk for both IBS constipation and IBS diarrhea. And we can see that effect size, an odd ratio of 4.24, that's large, with a nice significance there. So really important when we're wanting to address inflammation, we want to subdue this pro-inflammatory cytokine. How do we do that? There aren't many things that we have found, or when we were looking at the literature, that can directly influence this TNF-S15. But what we do know is that VEGF downregulates this pro-inflammatory cytokine. So what is VEGF? For those of you who have done DNA core or DNA sport and you're in the sports scene or actually just overall health, your VEGF encodes your vascular endothelial growth factor. We actually test a variant on this gene. And if you've got the variant, you may actually have increased expression of this growth factor, which is beneficial to sports performance, helps with fitness. But it also, if you've got good expression of this gene, it helps with down-regulating this pro-inflammatory cytokine. So how do we increase VEGF? That's how we're going to target this guy. We can do aerobic exercise, which really helps with the growth of this gene or the expression of this gene. And then certain proteins found from legumes help with the expression of VEGF. So of course, we want the protein and the clean protein from this only, but something like a pea protein, really nicely sourced, will help to increase expression of VEGF and then thereby down-regulate this pro-inflammatory cytokine. In terms of follow-up testing, we put in the intervention. We're wanting to again look at your hsCRP IL-1 levels. That would be really important. And then other stool tests, including those that look at calprotectin as a marker of inflammation. Then moving on to our serotonergic pathway, and this is where we're looking at your 5-hydroxytryptamine receptor, 3E. So this receptor controls gastrointestinal function to a large extent, not the only thing, of course, but it has a very strong role to play here with regards to peristalsis and secretion. We have seen that antagonists actually, and researchers, when they were looking at antagonists for this receptor, that it's been proven to be beneficial in the treatment of IBS diarrhea as well as in comorbid psychiatric conditions. And so there were certain drugs developed that actually block this receptor. So what we've also seen is it's relevant in emotional processing, mood, and visceral perception. And serotonin is obviously associated, or lack thereof, or too much associated with anxiety and depression, and disorders that are frequently comorbid with IBS. This specific receptor subunit is exclusively expressed in your gastrointestinal tissues. So this is where you will find it. You won't find it in the brain, exclusive to your GI tract. including your colon and small intestine. And it may play a distinct role in the formation and function of these receptors in the GI tract. So we're looking at a variant that actually leads to, it impairs expression and regulation and causes thereby upregulation of receptor expression. So it's kind of this backward forward mechanism, but at the end of the day, what you need to just keep in mind is there's going to be upregulation of a receptor expression, and that is going to in your colonic mucosa, and this is then a marker of an increased risk for developing IBS diarrhea. And it's because when that serotonin attaches to that receptor, it's actually working harder, and so there's going to be increased transit time with the receptor variant. So we see also very nice studies. This is quite a not so recent, but more recent ones as well, showing significant association between female patients with IBS diarrhea and the SNP, and a positive expression rate of this gene in patients with the GA genotype was significantly higher than in patients with the GG genotype. So a very strong association with IBS diarrhea-related symptoms. So now what we're wanting to do is find something that will act like an antagonist. We're not going to, if we don't have the drugs, let's look at other lifestyle and dietary factors that also antagonize this receptor, so kind of block it or down regulate it. Stress management, of course, there's specific probiotics, your Bilardi, Lactobacillus strains, Plantarum, Acidophilus, Rhamnosus, Infantis, from your Bifidobacterium strain that are really beneficial in overall IBS, but also trying to antagonize this receptor a little bit. More generalized is looking at a low FODMAP diet and going accordingly with that. And then specific phytoconstituents that antagonize this gene include your Panax ginseng, your Lingisticum striatum, and then a lot of your extracts from ginger. So things like gingerols, shogal's, ganna-lactone are all really good from ginger at actually antagonizing this receptor. Menthol, certain cannabinoids, and quinines have been found to also antagonize the receptor. Of course, I'm just listing the medication that you might see that your patient or your client would be on because of an antagonistic mechanism. And then also to note that there are certain foods that may be higher in serotonin. Your nuts, fruit, specifically bananas, tomatoes, and plums that agonize the receptor, and you might want to consider avoidance of those. Then with regards to follow-up testing, consider markers of stress response from your metabolomic or organic acid tests. And that is something you could look at from this side. Then moving to your starch degradation, which is looking at your enzyme, your sucrase isomaltase. I've got a nice little image here to show you your sucrose that as a part of the starch that needs to be digested, and your sucrase isomaltase that comes along and breaks that down so that we can have absorption processes beginning and everything working quite nicely. So we see obviously starch, this so this enzyme is a disaccharidase and a glycoprotein that is localized to the brush border membrane of your small intestinal villi. So keeping in mind, if there's already disruptions in your brush border, and I mean, inflammation can take those nice little fingers and shut them off, or they kind of get broken down or go away quite quickly. And things that enzymes that live in your brush border can't live there if your brush border villi are damaged. And when they become damaged, it's often with gastroenteritis, things like that, you can have damage to your brush border villi. So when you've got your enzymes living there, that's what you want. So you may have genetic variation that is leading to the enzyme not working well, but it might be not because of genetic variation that that enzyme is also not present because there's no way to live for short amounts of time, and that is potentially due to inflammation, gastroenteritis that can damage the villi of your brush border. Side point. So your isomaltase cleaves these branched dextrins, and your sucrase, part of this enzyme, digests your sucrose, your maltose, your short 1,4-linked glucose oligomers, and some of these branched starches into sugar monomers for intestinal absorption. How we found out about this specific enzyme and genetic variant is that there are individuals who carry a mutation on this enzyme, and when you see that in some of the literature, it will be known as an SI deficiency, where they completely do not have the enzyme that works. What we're looking at is not this complete mutation that completely shuts down the enzyme. It is a variation that decreases enzymatic function. Okay, so you still have function, it's just not as good. So we see deficient enzymatic properties resulting in a 35% reduced disaccharidase activity, and that is with your genetic variation. So not a complete deficiency and mutation, but a reduced efficiency to break down your starches. And that may lead to symptoms that would be considered your IBS, but potentially it's just because you have this enzyme that's not working that well when you eat a high starch diet. So with personalized support, look at reducing dietary intake of sucrose and starch, because then there's less for this enzyme to do. Potentially look at an elimination and a challenge diet. And then follow-up testing would be potentially looking at hydrogen breath tests that would be able to or give insight into carbohydrate malabsorption, and other comprehensive stool analysis that detect the presence of SIBO, also giving insight to starch, excuse me, breakdown with starch. Sorry, long lecture. So then moving on to gluten intolerance, and this is not IBS, but it's something that with many gastro or medical organizations like NICE in the UK, they recommend testing for celiac disease in individuals being evaluated for IBS. And this is because many, especially mainstream, it is celiac disease is considered a childhood disease oftentimes, and it can go a very long time without being diagnosed properly. We are seeing from the literature more and more that celiac disease is being diagnosed in adulthood into the elderly, and so really important that we screen for it if we can. We know that untreated celiac disease can lead to long-term health complications, so it's crucial to rule out in individuals suspected of having IBS. Here we're looking at gene products of your MHC complex, which are termed your human leukocyte antigens, or HLAs, and they function by binding and presenting peptides, including your gluten peptides, to your CD4 and your T cells, which are essential for initiating an immune response that then damage the intestinal lining in celiac disease. So if you carry these risk genotypes and then you are exposed to other viral infections, it can be if there's a higher family history of celiac disease, type 1 diabetes, certain individuals are more prone. And then you are exposed to gluten. So it's again, many things that contribute to the risk. But if you carry these risk genotypes and you take in gluten, or you're exposed to gluten, there is going to be this immune response. And depending on how severe, it can lead to this damage and then causing celiac disease. So your HLA DQ2, DQ8 genotyping is part of the diagnostic guidelines, especially in unclear cases. and very useful in individuals who are not on a high gluten diet to start off with, because many of those antigens might not be detected. So here is an image illustrating what I've just spoken about, where we have these gluten peptides that when they are exposed to our gastrointestinal tract, there is an immune response that happens, and we have the production of antigens, and that then leading to almost an autoimmune reaction to whenever we're exposed to gluten, or an inflammatory reaction. So when we carry the DQ2, DQ8 genotype, even if it doesn't develop into celiac disease, there are still going to be antigens that are produced. Even if it doesn't develop into celiac disease, there are certain antigens that are produced that you still may experience or may not even see it symptomatically, but there is within your body a pro-inflammatory response because of the genotype. So that is something to always keep in mind with this DQ2, DQ8 genotype. So will not always result in celiac disease, but may always result in some kind of level of an antigen being produced. For personalized action points, a gluten-free diet or challenge, and then testing for intestinal barrier permeability, as well as your anti-gliadin IgA. So those are some of the genetic variants that I would consider looking at if you are seeing someone presenting with IBS, symptoms of IBS, or you screening for other conditions, but they are coming in with gastrointestinal symptoms that are related to IBS, and then you are able to get more insights. Is it that? Is it something else? And how can I, together with functional testing, be able to target treatment and get the best results for the client? So with that in mind, again, just to emphasize who should be doing this test: anyone who might be suffering from, predisposed to, complaining around your altered immune function, compromised gut barrier integrity, gut dysbiosis, IBS, your inflammatory bowel conditions, skin conditions such as eczema, because that's again inflammatory, very strongly related to the gut, mental health, intolerances to certain foods that they're not always intolerant to, celiac disease, as well as your non-celiac gluten sensitivity, and those experiencing any gut-related condition. But very importantly, for those who are wanting to look at a healthier diet, healthier lifestyle to improve health, not just longevity, but improving health and health span. So that is where I would say DNA gut is a really important key test within your practice, and tests and paired with other functional tests. So not to look at in isolation, but to be paired with other tests. And I've listed a few, but there are many others, such as your GI map, your GI 360, other stool test offerings, OMX, SIBO tests. For babies, if you're looking at babies, it would be also useful to do, because especially when we're looking at immunity, inflammation, and your FUT2 genotype, really important in gastroenteritis and building a healthy bifidobacteria colonization, and that's where tiny health, your baby gut, would be useful alongside it. Your OAP toxin tests, because that would influence your detoxification, and many more, but very much considering this alongside functional testing for a targeted, personalized approach to your clients. As a reminder, and we are going to get into more of the detail around DNA gut, but also how it pairs and how it works within the functional healthcare approach, is where we're going to talk about it at the Nordic Conference, and some really great speakers and topics that will be discussed. So thank you very much.
Oof. That was a lot, Helen. Well done for getting all this information into
Yeah.
like
Well,
just over an hour.
I need another coffee.
Yeah, absolutely. But I think I just really want to say that we are beginning here. We are going to offer you more education on this test. The DNA gut education is already in our shop, so you can go in and purchase that. You also get a test and the education there. So start familiarizing yourself. I hope a lot of you realize that here we are going to really get additional knowledge to the stool tests that we already have, or the SIBO tests that we are using, but and intestinal permeability and all that. But here we are getting additional information, as sometimes we have complicated patients, not just sometimes, we often have complicated patients. They don't necessarily respond just like that. And we need knowledge. We need all this, all the pieces in the puzzle in order to understand why a patient is reacting the way they are. I especially like the area that you did on IBS and the inflammation, how IBS and also how the inflammation also can occur and is a part of the IBS picture, as well as gluten can be, and the DQ2 and the DQ8. So I know we are working on a document that where we're kind of pairing the GI map with the DNA gut test to give you clues as to which markers are intertwined, and we may even do a webinar on that at some stage. But hang in there with us, and yeah, follow the education that we are offering so that we can help our patients get optimal gut health. Really, that's what it's about, to do health optimization out there. So Helen, thank you very much, and I hope to see you all in London, of course, all 61 countries in London. We will share the slides and the recording with you, and also the link to the education and what else we are developing throughout the next period of time. We will keep you posted for sure. Thank you, Helen, and thank you all out there. Have a nice evening or day or night, wherever you are. Bye.
Bye.
If you do not already have an account with us, please contact our team and we will assist you. If you found this webinar helpful, please like and subscribe to our channel.